Good morning, good afternoon, good evening, wherever you may be watching us today. This is a brief lecture on the different parts of the cell. This is the first of a series of lectures on basic human histology. Our main reference is Junqueira's histology textbook and Netter's Atlas of Histology. Majority of the images we will employ are derived from the latter. The cell is the fundamental structural and functional unit of all living organisms. The body contains about 60 times 10 raised to the 12th, or 60 billion cells, of which there are approximately 200 different types. Cells vary widely in size and shape. A typical cell has polarized compartments and surface specializations. Internal cell structure is modified to reflect function. The centrally placed nucleus, labeled here as number 6, is surrounded by endoplasmic reticulum. Mitochondria occupy the basal compartment, here labeled as number 5, and the apical compartment, or the superior-most compartment, contains the Golgi complex and a centriole. Apical microvilli increase the plasma membrane surface area for absorption. The different parts of the cell in this cross-section are as follows. Number 1 here, the centrioles. Number 2, the microvilli, singular is microvillus. The endoplasmic reticulum, which has a rough component labeled as number 3 here, the rough endoplasmic reticulum, or RER, and surrounding the rough endoplasmic reticulum is the smooth endoplasmic reticulum, or SER. Located again in the basal compartment is the mitochondrion, labeled as number 5. Centrally located in a typical cell is the nucleus, and surrounding the nucleus, labeled as number 7, is the Golgi complex. What follows is an electron micrograph of a gap junction in cardiac or heart muscle at low and high magnification, and below it is a schematic of a gap junction. Metabolic, ionic, and low resistance electrical communication occurs between adjacent cells via gap or communicating junctions, in which a narrow gap of about 2 nanometers separates opposing cell membranes. Gap junctions are specialized sites composed of large, tightly packed intercellular channels which connect cytoplasm of adjacent cells. Each cylindrical channel, such as labeled here, is 10 to 12 nanometers long and 2.8 to 3 nanometers in diameter, consisting of a pair of half channels. The half channels are called connexons, and these are embedded in the cell membranes. Each connexon comprises 6 symmetric protein subunits called connexins. The connexins are transmembrane proteins surrounding a small um, central aqueous pore, and several diseases can actually result from mutations in genes encoding connexins, which are named according to molecular size. For example, recessive mutations in connexin 26, let me write that down, connexin 26, with a molecular size of 26 kilodaltons, lead to the most common cause of inherited human deafness, which often affects the elderly. Connexin 26 is usually involved in potassium, transport in cells that support cochlear hair cells. So mutations in connexin 26, hearing loss. The different parts of the cell junctions are labeled as follows. Number one here is the plasma or the cell membrane. Number two, the gap or the communicating junction. Number three is a connexin monomer, the cylindrical tube-shaped structure over here. Four is the hydrophilic channel or pore. And five here is a connexon, which is a hexamer, meaning it is composed of 6 protein subunits. Shown here is the nucleus of a lymphocyte in electron micrography. The nucleus is the largest, most conspicuous structure in any cell, most cells at least. Most cells have only one nucleus. The nucleus consists of the nucleolus, chromatin, nuclear matrix, and a nuclear envelope. A nuclear envelope, labeled here as number 2, encloses the nucleus, shown here in the big ovoid structure in the center. The nucleus of interface cells and separates the nucleus from the cytoplasm. The nucleus contains genetic material, which is DNA, that is either packed or unpacked. Heterochromatin, shown here as the densely colored structures in the center of the nucleus, is the packed form, whereas euchromatin is the unpacked form. Therefore, euchromatin is unpacked, heterochromatin is packed. Histone proteins are involved in the packaging of DNA into heterochromatin. Euchromatin represents unwound DNA in the process of transcription. The proportion of heterochromatin to euchromatin gives an indication of the general activity of the cell. Mature erythrocytes, which are red blood cells of most mammals, do not contain nuclei since they are extruded during development. Histopathology uses changes in nuclear morphology as diagnostic features. Many cellular disorders show an increase in nuclear to cytoplasmic ratio, nuclear indentation, ground glass appearance, crystalloid inclusions, or abnormal multinucleation. Aberrant nuclear location in a cell may also indicate cellular pathology, such as in the presence of centrally located nuclei in skeletal muscle fibers of patients with muscular dystrophy. Shown here is an electron micrograph of a nuclear envelope, labeled here as number 1, and a schematic of a nuclear pore complex, or an NPC. A nuclear envelope encloses the nucleus of interface cells and separates a nucleus from the outer cytoplasm. The nuclear envelope consists of two parallel unit membranes. This one, you can follow my arrows over here. Two parallel unit membranes separated by a narrow space, roughly only 10 to 70 nanometers wide, which is the perinuclear space or the cisterna. Many small octagonal apertures called nuclear pores, labeled here as number 2, nuclear pores, perforate the nuclear envelope. About 100 nanometers in diameter, they permit selective, bidirectional exchange of small molecules, ribosomal subunits, and other substances between nucleus and cytoplasm. The outer rim of each pore forms by fusion of outer and inner nuclear membranes. An NPC, spanning the opening of each pore, consists of 8 proteins. This is a schematic of an NPC, or nucleoporins around a central plug or a granule. The number and distribution of nuclear pore complexes vary widely according to activity and type of cell. Nuclear pore complexes are especially numerous in metabolically active cells. Genetic mutations in the nucleoporin alanine, let's just write it down here so that we remember. Genetic mutations in the nucleoporin alanine have been linked to the autosomal recessive AAA syndrome, also known as Algrove syndrome. Algrove syndrome. The other name for Algrove syndrome is a mouthful. It's achalasia, addisonianism, alacrimia syndrome, which is why it's called the AAA syndrome. To recall, genetic mutations in the nucleoporin alanine are linked to the autosomal recessive AAA syndrome, also called the Algrove syndrome, which is lengthily called the achalasia, addisonianism, alacrimia syndrome, AAA syndrome. This is a schematic of mitochondria, which are, of course, the powerhouse of the cell, of any cell. And follows is an electron micrograph of the same mitochondria in a hepatocyte or liver cell. Mitochondrial shape varies with the plane of section and the type of cell. Each organelle has a thin shelf or tubular cristae. These are the cristae, which are tubular and folded, which project into the mitochondrial matrix here, labeled as number 3. The mitochondrial matrix is the lighter colored substance of the inner mitochondrion. Each mitochondrion has thin shelf or tubular cristae projecting into the matrix. The outer mitochondrial membrane is a smooth contour. It's shown here. It's like the wrapper of a sausage or a hot dog. It consists mostly of a large channel-forming protein called porin. Porin increases membrane permeability for passage of molecules and metabolites required for ATP synthesis. And the inner mitochondrial membrane, as we have discussed, this one over here, is thrown into a series of folds called cristae, which project into the mitochondrial matrix, which has an increased electron density, making it finely granular. This is an electromicrograph of part of an active fibroblast and higher magnification electromicrograph in the lower pane of part of a protein synthesizing cell. And shown here are the ribosomes. Ribosomes are small, spherical, electron-dense particles that synthesize proteins. They are uniform in size, about 15 to 20 nanometers in diameter, and they consist mostly of RNA and associated proteins. We have two kinds of ribosomes, free ribosomes and polyribosomes. Free ribosomes, shown here in number 2, occur in the cytoplasm, and they are single particles or rosette-like clusters. When the free ribosomes form clusters or groups, such as in number 3 over here, they are then called polyribosomes. And polyribosomes consist of several ribosomes arranged along a thread of messenger RNA or mRNA. Single ribosomes are inactive, whereas polyribosomes are the ones that are active in protein synthesis, where they assemble specific amino acids into polypeptides. Ribosomes may be attached to membranes of the rough endoplasmic reticulum or RER or to the outer nuclear membrane. Polyribosomes also synthesize proteins for internal use only, for internal use by the cell. It is the free ribosomes attached to the RER that engage in protein synthesis for export from the cell or for proteins destined for lysosomes, which are organelles for um, disintegration and recycling of cellular waste. This is a schematic and a high magnification electromicrograph of the Golgi complex, showing its different functional compartments. Shown here in number one is the trans surface of the Golgi complex. More innerward is the saccule of the medial compartment, the saccule. Still in an inner compartment is the cis surface of the Golgi complex over here. And finally, out in the outward compartment are the different Golgi vesicles. The Golgi complex or apparatus is a complex array of flattened, slightly curved, closely packed membrane-bound sacs. This highly polarized, compartmentalized organelle has convex and concave sides and three functionally distinct compartments. There is the cis Golgi network of vesicles on the convex side. There's a medial compartment of sacs of flattened saccules, also known as vesicles. And on the concave side of a trans-Golgi network of vesicles and vacuoles for distribution and sorting of secretory products. Each saccule in the medial compartment contains a different group of processing enzymes that reside in its membranes as integral membrane proteins. Some cells have one Golgi complex. Others, which actively synthesize proteins and polysaccharides, have many. The Golgi complex adds proteins to sugars to form glycoproteins, assemble polysaccharides, elaborates membrane lipids, and produces lysosomes that are kept by cells. This assembly of the Golgi complex occurs in cells undergoing apoptosis or programmed cell death, whereby extensive and irreversible fragmentation of this organelle forms small tubulovesicular components. This feature is probably due to a caspase-related cleavage of integral Golgi tethering proteins. Caspases are proteases in the cytoplasm that become activated early on in apoptosis. Caspases. We now take a closer look at the bulk of the substance of the cell, the cytoplasm. The cytoplasm is a complex aqueous gel made of water. Around 70% of the cytoplasm is water, and the remaining 30% are proteins, lipids, carbohydrates, and organic and inorganic molecules. Organelles and inclusions are all in the cytoplasm. Electron-dense glycogen particles within the cytoplasm form aggregates and are not membrane-bound. Peroxisomes, labeled here as number 5, so this creature over here, peroxisomes are enveloped by a single membrane and they contain crystalline or strand-like deposits. The mitochondria, labeled here as number 3, these are the mitochondria. Let's put them in boxes. The mitochondria are distinguished from peroxisomes because they have an inner and an outer membrane. The peroxisome only has a single membrane. The Zellweger or the cerebrohepatorenal syndrome is a peroxisomal disorder that usually leads to abnormalities in the brain, the kidneys, and the liver. The Zellweger syndrome, a peroxisomal disorder. Affected infants die soon after, which is most likely due to faulty neural cell myelination in utero. This is a high magnification electromicrograph of the glycogen rosettes in a hepatocyte. Glycogen, which is a cytoplasmic inclusion, appears as small electron-dense particles. They are free in the cytoplasm and glycogen are not membrane-bound. So these are glycogen rosettes, grape-like clusters over here. Aggregates of glycogen particles form irregular patches known as alpha particles or rosettes. Glycogen, which is a D-glucose polymer, is stored mostly in the cytoplasm of liver cells and skeletal muscle fibers. It also occurs in smaller amounts in other tissues. Synthesis, storage, and breakdown of glycogen occur rapidly according to need. Glycogen storage diseases, or GSDs, are a group of more than 10 inherited inborn errors of metabolism. They affect the synthesis or breakdown of glycogen. These are autosomal recessive disorders, typically occurring in infancy or childhood. As an example, von Gierk's disease, let's spell it out, von Gierk's disease, or type 1 GSD, is a deficiency of the enzyme glucose-6-phosphatase. Glucose-6-phosphatase deficiency leads to an abnormal accumulation of glycogen in muscle, liver cells, which causes clinically significant end organ disease and morbidity. Von Gierk's disease, also known as type 1 glycogen storage disease, caused by a deficiency in glucose-6-phosphatase enzyme. This is a closer look of a cavulae and vesicles in the endothelial cell of a capillary. Cells have several kinds of membrane-bound vesicles that form by invaginations of the plasma membrane, also known as the cell membrane. They then enter the cytoplasm by pinching off from the surface and are transported to other parts of the cell. Many cavulae, here all labeled as number 1, many cavulae in endothelial cells mediate transcytosis. Transcytosis, which is the process by which vesicles derived from cavulae are taken across a cell and release their contents at another surface. Other kinds of cytoplasmic vesicles, most derived from the Golgi complex, engage in exocytosis. Vesicles move to the cell surface, they fuse with plasma membranes, and they discharge contents to the cell exterior. Familial hypercholesterolemia is an autosomal dominant disorder caused by a mutation in the gene on chromosome 19. Cholesterolemia, over here, an autosomal dominant disease caused by a mutation in chromosome 19. Chromosome 19 gene here encodes LDL receptors, which are the receptors for quote-unquote the bad cholesterol. The defective receptors lose their affinity for coated pits and cellular uptake of cholesterol is blocked. This in turn causes a severe elevation in serum cholesterol. Hypercholesterolemia, a deficiency, a mutation in chromosome 19 gene that encodes the LDL receptors. Finally, an electromicrograph of microtubules in a cultured cell. Microtubules, labeled here as number 1, these are hollow, semi-rigid cylindrical organelles of uniform diameter, around 25 nanometers, but are extremely variable in length and are particularly abundant in neurons, platelets, leukocytes, and dividing cells. They form a major component of the cytoskeleton by helping to provide mechanical strength and establish cell shape. They engage in intracellular transport of organelles, ciliary and flagellar motility, mitotic spindle formation, chromosome translocation, and cytokinesis during cell division. They are not bound by a membrane, and their walls are composed of linear polymers of the globular protein called tubulin, hence the name microtubules. Microtubules are also the main constituents of cilia, flagella, and centrioles.